Alright, guys, so in this podcast, we're going to take a look at the third and final step of cellular respiration. And this step is what we talk about as oxidative phosphorylation. Now, just like step 2, this one's got multiple mini steps that make up the big picture. So, we're going to talk a little bit about your electron transport chain, which is what's going on right here, and then the final piece, which is the chemoosmosis, and that's right here. Now, if we take a look at this word oxidative phosphorylation, remember you've seen the term phosphorylation before. We talked about that in cell transport, where we're taking a phosphate off of ATP. As a result, we end up creating that ADP, which is right here, and adding that phosphate to something else. Well, in this case, rather than adding that phosphate to, say, a protein in your cell membrane, where it changes its shape and releases things to the opposite side, like your sodium-potassium pump, now we're going to take a series of chemical reactions, oxidative reactions, which is what's going on here. Oxidative you'll come across if you ever take AP chemistry. But we're going to use what the electron transport chain is doing to now add that phosphate rather to a protein. Now we're going to add it back to ADP so that we can create ATP in that final step. So let's take a look at each of these pieces of step 3 in a little more detail. Alright, so if we focus on your electron transport chain, which is really sort of the first half of oxidative phosphorylation, remember that back in glycolysis in your Krebs cycle, and even in that grooming step that links those two, we were creating those high energy electron carriers, the NADH and the FADH2. The whole purpose to creating these electron carriers is to actually use them here. So we go through glycolysis to break down glucose. We go through the grooming to make acetyl-CoA. We go through your Krebs cycle using that acetyl-CoA, and its whole purpose up until this point is to be creating these electron carriers because we need them here in this final step. Why do we need them? Okay, well, here's the deal. So we've got a bunch of these enzymes, okay, which are these purple things, these electron carriers that are sitting here in our inner mitochondrial membrane. So here's our mitochondria. Remember I said that we had all of those cisternae, or those folds, in that inner membrane of the mitochondria for this reason. We wanted lots and lots of surface area in that membrane so that I can fit lots and lots of these protein complexes, these pumps, into that membrane. The more surface area I have, the more space I have to add these proteins. And what we're going to do is remember that we added electrons and hydrogens from NAD plus to create NADH and from FAD to create FADH2 in glycolysis and in your Krebs cycle. Well, here we're going to basically go backwards. We're going to recreate that NAD plus and that FAD by taking those hydrogens off. Where are they going to go? Well, they're not just going to go floating off into space. We're going to use these proteins as pumps, just like your sodium-potassium pump. Now I'm going to pump hydrogens to this inner membrane space, so this space in between the outer membrane and the inner membrane of your mitochondria. And what that's going to do is create a concentration gradient. We've got low concentration of H plus ions here and a high concentration of H plus ions here. Now it's going to take energy to do this because I'm constantly trying to keep a concentration of H plus high in here. So not only am I pulling the hydrogens off of these electron carriers, but I'm using the electrons that are associated with them to keep providing me energy to make that concentration gradient. What that's going to do then is allow us to have a concentration gradient sitting in that inner membrane space.
So something to keep in mind while we're talking about the way that this transport system is working is that because I'm using energy from the electrons that I'm pulling off of these electron carriers to make these pumps work and creating a concentration gradient as a result of taking those H pluses across that membrane, this is active transport. I'm using energy to maintain a concentration gradient. The other interesting thing here is to note what I'm creating in terms of that gradient. So I'm using these H plus ions, and it's really important to understand the fact that it needs to be an ion and it can't just be hydrogen atoms, right? So remember, an ion means that I have a positive or negative charge because I've either added or removed electrons. In this case, I've removed electrons from a hydrogen atom. That's why they're H plus, right? Removed a negative. As a result, I have more positive charges in the nucleus of that atom, hence the positive charge on the hydrogen. And the really important part here is to remember that this is a membrane, right? These little guys are those phospholipids. So just like we talked about what kinds of things can move easily across your cell membrane, the same properties apply to any membrane that is made of phospholipids. And in the case of this inner mitochondrial membrane, it's a phospholipid bilayer just like your cell membrane is, which means anything that would be nonpolar, aka would have no charge, could easily diffuse right back through that membrane. But in this case, we are using ions, right? So ions have a charge, which means they are not going to interact with the fatty acid tails because they're different, right? Those fatty acid tails are nonpolar, and anything that has a charge, so polar or an ion, can't pass through them. So that's really important. So not only are we using the electrons I'm stealing from those hydrogen atoms to make these pumps work because I need to create a concentration gradient, I need energy to do active transport here. But those H plus ions then are also stuck out here. They can't easily get back through this fatty acid tail layer in this phospholipid bilayer, which means I can now force the concentration gradient to exist. And that also means I'm going to couple that with this process we'll talk about a little later called chemoosmosis, which is essentially a passive transport system. So I'm going to use those H pluses and they're going to diffuse through this protein to ultimately create all that ATP. So by having those be ions, they can't just pass through this membrane wherever they want to. They are forced to go through this protein here, which is what your cells are ultimately trying to accomplish. So if for some reason there was some sort of mutation or damage done to that mitochondria where there was a hole taken out of this membrane, then those H pluses could pass through here um, and as a result not be forced necessarily through this protein, which would end up decreasing your ATP production, which is a really important process because this is how we're going to produce the vast majority of the ATP that we get out of cellular respiration as a result of doing this process and having that concentration gradient in place. The other component here is that those electrons are ultimately going to get passed down and attached back to H pluses that are hanging out here in the matrix. And we're going to attach that to oxygen, which is why oxygen is so important. It's going to be our final electron acceptor to create that water. So by using these proton pumps, we get all of this concentration gradient to exist, and to ensure that we keep high here, low down here, we also then attach those hydrogens to oxygen when they diffuse back through this membrane and create the water. And so that also keeps the concentration low back down here as well.
Okay. So energy gets released from the enzymes themselves, the enzymatic reactions that they're catalyzing, and now I have this hydrogen gradient sitting here. Okay, so remember, high in that inner membrane space, low inside the matrix. All right, and again, remember that we've got all of those cristae, those folds in that inner membrane to give me the extra surface area to make sure that I've got lots and lots of these proteins, lots and lots of these enzymes that are associated with the electron transport chain. Okay, so that's the electron transport chain in and of itself. It's just pumping hydrogens into our membrane space to create a gradient. Okay, and we're using those electron carriers that we got from glycolysis, from the grooming cycle, and from the Krebs cycle to have those electron carriers here to carry those H pluses with them. All right. The final piece to this is down in here, okay? Now that we've created this gradient by using energy, by making these proton pumps work, now down here we're going to allow diffusion to happen. And what's going to happen is that these H pluses, now that I've created this gradient, remember high here, low down here, those H plus are going to diffuse through an enzyme called ATP synthase. Remember, ASE tells me it's an enzyme. And this ATP synthase is really the major enzyme sitting in your mitochondrial inner membrane that works for chemoosmosis, which is the second half to oxidative phosphorylation. Okay? And what we end up doing is here's where oxygen's needed. Right? We got to breathe in oxygen. Here's why. Without oxygen, there's nothing to pick up those hydrogens that I'm shuffling along this electron transport chain and down through ATP synthase. They have to have something to attach to. And so what do they attach to? They attach to oxygen, and that's where we get some of the water out of cellular respiration. What is this actual enzyme doing though? Well, every time we get an H plus to go through that enzyme, right, as it diffuses through, this spins like a rotor. All right, so for every H plus that comes down, this clicks around once. Every time that spins, we're taking ADP plus a phosphate, giving it the energy that's necessary so that they can hook back up and create ATP, which again is the ultimate reason we're doing all of cellular respiration in the first place.
So chemoosmosis is going to take advantage thus of having this concentration gradient here. Because they're ions, they're forced to go through that ATP synthase enzyme in order to diffuse. So this really is an example of not just osmosis or diffusion being that passive transport, but really it's facilitated diffusion, right? So we're using the fact that that electron transport chain has created this concentration gradient through active transport, and then we're going to couple that with facilitated diffusion of those H pluses through that ATP synthase enzyme to create all of that ATP.
How do you turn that bite of food into a chemical that a cell can recognize and use as energy? The first step is altering the food into its component chemical compounds and then getting those molecules into your cells. That process is called digestion. Once inside your cells, the process of turning that bite of food into useful energy by cellular respiration begins. The process of digestion results with carbohydrates and other molecules being removed from the consumed food and transported into the bloodstream. From there, nutrients, like the carbohydrate glucose, will leave the bloodstream through a capillary wall and enter a tissue cell. Once inside the cell, cellular respiration will completely oxidize the glucose molecule, releasing high-energy electrons. The overall goal is to make ATP, a storage form of energy for most cells. Cellular respiration is a four-stage process that begins with glycolysis. Glycolysis literally means splitting sugars, and it is the first step of cellular respiration, occurring in the cytoplasm of the cell. Glycolysis consists of two distinct phases, an energy investment phase and an energy harvesting phase. In the energy investment phase, two ATP molecules transfer energy to the glucose molecule, forming a 6-carbon sugar diphosphate molecule. This molecule splits, and the energy harvesting phase begins. During this phase, the two 3-carbon molecules are converted to pyruvate, and ATP is formed. Glycolysis is a 10-step reaction that involves the activity of multiple enzymes and enzyme assistance. In the process, a net of 2 molecules of ATP, 2 molecules of pyruvate, and 2 high-energy electron-carrying molecules of NADH are produced. When oxygen is present, the pyruvate molecules and NADH enter the mitochondria, and the next stage of cellular respiration begins. The next stage of cellular respiration involves the movement of pyruvate into the mitochondria, where it undergoes oxidation. Each pyruvate molecule is converted into a compound called acetyl-CoA. In the process of pyruvate oxidation, electrons are transferred to NAD, producing NADH, and a carbon is lost, forming carbon dioxide. The next stage is the citric acid cycle, also called the Krebs cycle. Here, acetyl-CoA will bind with a starting compound called oxaloacetate, and through a series of enzymatic redox reactions, all carbons, hydrogens, and oxygens in pyruvate ultimately end up as carbon dioxide and water. The pathway is called a cycle because oxaloacetate is the starting and ending compound of the pathway. For every glucose that enters glycolysis, the cycle completes twice, once for each molecule of pyruvate that entered the mitochondria. During pyruvate oxidation and the citric acid cycle, a net of 8 NADH, 2 FADH2, 2 ATP, and 6 CO2 are produced for each glucose molecule. In order to understand how the majority of the energy is produced by aerobic respiration, we need to follow the NADH and FADH2 molecules to the next stage, the electron transport chain. The electron transport chain is a series of membrane-bound carriers in the mitochondria that pass electrons from one to another. As the electrons are transferred between the membrane proteins, the cell is able to capture energy and use it to produce ATP molecules. Proteins in the chain pump hydrogen ions across a membrane. When the hydrogen ions flow back across the membrane through an ATP synthase complex, ATP is synthesized by the enzyme ATP synthase. Oxygen acts as the terminal electron acceptor. By accepting electrons, oxygen is reduced to form water, a byproduct of the electron transport chain. All the high-energy electron carriers from the previous stages of cellular respiration bring their electrons into the chain. From this, the bulk of ATP from the entirety of cellular respiration is produced, a net of 32 to 36 ATP. In summary, we have seen how the 4 stages of cellular respiration are responsible for converting the energy found in the glucose molecule into ATP, the energy battery of the cell. On average, 36 ATP molecules are produced per glucose molecule that entered the cell. In the process of producing ATP, oxygen is brought in from the bloodstream to be the final electron acceptor in the electron transport chain, and the carbon dioxide that is produced as a byproduct is released. The goal of cellular respiration is to transfer the energy from the food that we eat daily into ATP that our bodies can use. This process starts with the eating of a snack or meal and ends with capturing the energy from the complete breakdown of the nutrients into energy and carbon dioxide.